sequences, protein sequences, RNA sequences. So whatever the sequencing result we can analyze by the bioinformatics tools. So we can start from the basic tool, that is BLAST, that is sequence similarity search tool. So it is working. This tool is working online as well as offline both. So most of cases, the student use the online version of this basic local alignment search tool, and how to use it offline and how to install in your system and how to run, we can discuss it today. So basically, local BLAST or standalone BLAST is used for the homology homology finding from different sequences using DNA, RNA, and protein sequences. So why it is used? It is actually basically it is used to identify the unknown sequences which we have not known about the sequences. So we can run the BLAST with known sequences, and we can easily identify with the unknown sequences. And second one is the build a homology tree. So if we want to check the homology tree about different species, we can also use this tool. And after that, we can identify the protein structure by aligning the sequence of the protein with the known structures, and we can easily identify the unknown protein which belongs to which other known protein structures. And after that, we can use it for map the sequences in a genome. And likewise, there are many applications of BLAST. So we start the local install, how we can use this local install BLAST, and what are the advantages. So you can see by use of pointer, I can elaborate. So in this basic local alignment search tool, it has a advantage of you can search many input sequences at once. So in this locally installed BLAST, you can search 10, 20, 100,000, whatever you required, you can take as an input and run locally. So there is no limit for any sequence similarity. So you can take multiple sequences. And other advantage of the offline BLAST is customizable database. So in this customizable database, you can create your own database. Whatever you have research you have performed, you can take that database and you can search in that database. And the third one is the no need for internet access. Once you have to download and install this tool in your computer, and then after, there is no need for any internet access. And it is also a fast work faster and for small and medium sized databases. And another is the integrated BLAST search into a larger automated bioinformatics analysis. So start from this what required to run this basic local alignment search tool. So this is our FASTA format sequence, which is start from the greater than symbol. So this is the first requirement. So you can download the sequences from internet like NCBI and UniProt, etc., and use as query sequence. And you can see this sign, this symbol, it is greater than symbol, and it depicts that this is a sequence is in the FASTA format. So you can use it as a query, and in database also you can use the FASTA sequence. So how many types of BLAST? So BLAST is a collection of 5 programs for different combinations of query as well as query and database sequences. So first one is the BLAST N. So when BLAST N use when our query sequence is DNA sequence and our database sequence also DNA nucleotide sequence. And when we use the BLAST P, so BLAST P used when our query sequence in the protein and database also protein. And likewise BLAST X when our query sequence is nucleotide and our database sequence is protein. So then we use to BLAST X. Likewise, tblastn and tblastx in this when query sequence is protein and nucleotide sequence used for database. Likewise, this tblastx translated query sequence is the nucleotide sequence, and the database is also nucleotide, but these both are work after 6-frame translation. So most widely used blast, these three types of blast most widely used in the research. So this is the actual workflow of the blast. So part of the standalone blast, the blast program file downloaded from NCBI contains blast p, blast n, blast x, blast t, blast x, and make blast database. These files are there. So start from your query. You can input your query and select the appropriate program to run, and after that you can select the database which you have designed. By this way you can take a FASTA file and create your own database and use this database and align this query sequence using this appropriate program. Then you can find the output. So this is all about the basic about blast. So we can use it by hands-on module, and you can see if you have your computers or laptops, then you can also do the same work, and you can easy learn how to install and run the blast. So first of all, you can go to the site, this site, and this is the NCBI site. In this, different type of executable blast files are available. You can download among you as your computer. Like if you're using Linux, then you can use the Linux version of blast. If you are working with the Windows version, then you can use the Windows version of the blast. Likewise, these are the different setups available according to the computer. So once you download, you can install. You just double click on this tool, on this setup, and just install this tool. And after that, after installation, you have these two files. First one is the query file and second one is the database file. If you have these two files, then you can use these two files to run the blast. So the blast is installed in this directory. So here in bin folder, you can paste the database file as well as gene query file. So these are the command line important parameters. What are the input and output? What are the database? How we can give the command to run the blast? Here is the hyphen query. It is start from query name and dot FASTA. Specify the input file, and dot hyphen out is the specify for the output file, and this database which denote the database, and this out fmt is actually specify the what output type you want, and for more detail you can type the help. So these are the basic commands. So first of all, you can open the command prompt. After that, type the path of the bin folder of the blast where blast install. You can copy this path and paste in the bin folder. Then type cd space and the location of the bin where you paste the query as well as database files. After that, you can run this command to make a database. So suppose if you have a database sequences of 1000 or 2000 or 10,000, so this is the so this is the command. So this is the file name of database sequence. Okay, and this is this denote DB type. So if you are using with you are working with the nucleotide sequence of database, so here you can specify the NuCl if you want to create a database of nucleotide sequences. And if you want to create a database of protein sequences, so then you can specify prot. So kept in mind that these two commands are necessary to select. Okay. After creating the database, just run the blast type by blast command blast n hyphen query gene1.fasta hyphen database, and it is the database name dot fasta, and hyphen out is the result dot out. So these are the basic command you can type, and it will run and create your result. So this is the result file which have been created in your bin folder. So you can see this blast version, and this is the reference which use to cite when you are using the blast standalone tool. So after that, you can see the database uh, total sequences in database, total 9 sequences, total 9,000 total letters. It means there are 9,000 letters. And in query sequence, you can see the name of the query sequence, and the length of the query sequence is 988. So sequence producing significant alignment with this is the sequence present in the database, and you can see that the length of the database is 1000. So after aligning this sequence, these are the parameters which are generated, like score, value, and identity. It is 99% identical sequences, and the gap, how much gaps are inserted in after aligning, that is 12. And strand, these both are the plus strand. So this is the interpretation of the result, and this is the alignment. So here a query start from 1, and it is a database sequence start from 1. So these are the aligned nucleotide residue. So here is the output in the column format. So you can save it using out fmt command, which we have previously discussed. So this result, you can see there are total 12 columns, and what are the interpretation of each columns. First one is the query sequence of the query name, that is query sequence ID. And second one is the subject, which is in your database sequences. So database sequence ID, subject ID, which is known as. And the third one is the percentage identity. So percentage identity is the third column. So it is 98.8%. So after that, it is the length of alignment. Total length of alignment is 1000. So total number of mismatches in sequences, how much mismatches are there? 0. And total gap open penalties, how much places where gap opens? That is 1 place. And query start from aligning from 1, and query end at alignment, that is 988. So query align from 1 to 988, and the subject sequence in the database start from the align from 1 to end at the 1000. And after that, you can see the e value that is 0.0, and the bit score, that is 1770. So these are the parameters you kept in mind to analyze your result. So the result parameters, that is score, bit score, so should be higher for similar sequences. E value should be less than 0 or equal to 0 for more significant result. And identity, higher the identity shows higher the homology between sequences. And last one is the query coverage. How much query percentage of your query is covered by the database sequences. So these are the main parameter you should kept in mind when you run the blast locally or globally or your website. So these are the applications. Uh, like in disease diagnosis and pathogen identification. So identify the bacterial, viral, or fungal pathogen by comparing patient-derived DNA, RNA sequences against known database. So the good example of this is the SARS-CoV-2 COVID variant identification by aligning the sample with the predefined viral genome sequences. If there is much similarity, then you can say that this variant is belong to this variant. After that, you can see the another example in medical science, the genetic mutation detection. So detect disease-causing mutations such as SNP indels by aligning patient DNA or a reference genome used in cancer genomics, inherited disease analysis, and personalized medicine. So when align two sequences, if there are mismatch, then you can easily identify what are the SNP among those sequences. And another is vaccine development. So identify conserved protein or gene regions in the pathogen of vaccine target discovery. For example, epitope mapping in the influenza or COVID-19 vaccine. And likewise, the drug target discovery identify the validated homologous sequences, gene proteins as a potential target for the drug discovery. And as well as the microbiome analysis, BLAST is also used to classify the microbial species from sequencing data in gut or skin microbiome studies. So likewise, in medical sciences, there are many applications of this basic local alignment search tool in the agriculture sector also. In agriculture science, it is used for crop improvement and breeding by identifying the gene links to drought resistant, pest resistance or yield trait in crops, used for marker assisted selection and genomic selection programs. And it is also used to identify the crop is GMO, genetically modified or not. So confirm the insertion or integrity of the transgene in the genetically modified crop by aligning your prior sequences to the modified sequences. In the third one is the plant pathogen detection. So rapid identification of plant pathogen, virus, fungi, bacteria through sequence alignment. Example, identify rice blast fungus. So likewise, these are the applications in like comparative genomics. Compare the gene family between species like maize, rice, wheat, other likewise other species. You can find the conserved or divergent gene. And likewise, soil and rhizosphere microbiome studies analyze microbial diversity in soil to enhance soil health and improve plant microbe interactions. So these are the basic applications which are widely used blast. And you can, for more detail, you can use the you can go and visit the site of NCBI blast. So these are the basic steps. So let's start the hands-on module, how to run on your computer. So you can see this is the NCBI home page. So it is a NCBI is a database of bioinformatics related sequences as well as structure. So you can search your database here. You can select the query sequence, whatever you want. You can select. So I will discuss with the example of the plant lipoxygenase protein or nucleotide. So in this case, I am using the coding sequences. So these are the sequences: glycine max, Arabidopsis thaliana. So you can select the query sequences. Okay, so you can select 10 query sequences for your example, and from here you can download these in FASTA format, FASTA, and create file. So you can see these are the query sequences which you have downloaded from this FASTA file in the FASTA file format. So now you can use it as query sequence. So how to save these sequences? You can just put that FASTA at later, and here, from here you can select R type, and now you can save this. Okay, so if you select if you can you can select this file and check whether it is saved or not. You can find that these are the FASTA file formatted sequences. You can count how many files are present in your text file. You can count it by this one. That is total how much? Total 10 FASTA file sequences are present in your text file. So likewise, you can download your database. So it will show the total 50 sequences in this page. And now you can select other sequences as a database. So we have selected another 10 sequences to save as the database sequences. So save in the similar manner, select FASTA and create file. You can see this is the sequence of database. You can check, and now you can save it. All type, and here dot FASTA. Okay, so likewise you can save the query as well as database sequences. And if you have these two, so this is your query and this is your database sequences. So in this case, we are using 10 query and 10 database sequences. So for a smooth running, we can use 5 query sequences total. Okay, so now we are using 4 query sequences and to find out the sequence homology in 10 database sequences. So these 2 files are important to run the program locally. So after that, you can download the Blast standalone tool from NCBI database. Just paste this, or you can search standalone Blast. So this is the Blast Plus executable page. From this page, you can download by clicking on this. You can see this. These are the list of the setup which are available according to your computer. In our case, we are using this one because we are working on the window-based system. So just click on this and it will start downloading. So total size is 133 MB. So I can cancel this because I have already downloaded this tool. In the download folder, you can see I have downloaded this one, and after download, you can extract this setup by using WinRAR. It is freely available tool. You can extract the file. Okay, after extracting the file, you can see this folder will be created. Okay, and in this folder, there is a bin folder available. So as we discussed in our lecture, that we should place the query as well as database sequences in this bin folder. Okay, so this is the query sequence and this is the database sequence. So we just paste both our sequences in the bin folder. Okay, so you can see these are the 2 sequences, database sequence and query sequence. After pasting this sequence, you can see according to the manual. Open command prompt. So you can open the command prompt in your computer by typing CMD. Okay, you just click on this. Okay, so you can see this is the command prompt in the window. You can now you can paste the location where your query and database file are stored. So in our case, it is stored in bin folder. So we can copy this by this way. Just click one time and copy, and just paste by using cd command. So now you can see you are in the bin folder. So what are the files stored in bin folder? You can click, you can check it by typing dir. So these are the files which are stored in the bin folder. So where are your files which you have pasted in this folder? This one is the database sequence dot FASTA, and likewise query sequence dot FASTA. Okay. So after that, you can create the database which you have downloaded from. Uh, just paste this command. So actually, because we are using nucleotide sequences, so here we use the NuCl command in DB type, and here we just provide the name of the database. So in our case, it is DATA, database sequences dot FASTA. Okay. So I have provided the name of the sequence of database. After that, we have to create the database by typing this command and just enter. Okay. So you can see the database has been created total in the 0.23 second. So in database, in the bin folder, you can check these are the files which are created like database sequence.fasta.ndb. So these are the internal files which are created by the BLAST tool. After the creating of database, you can run this further. If you are using protein database, then you should use the prot in the place of NuCl nucleic acid. So now we are ready to run our program. Just copy this blast n minus query, and here you provide the query name, query sequences dot FASTA minus TB, and database sequence, database name minus out final result dot out. So by clicking this, you can see in your bin folder final result dot out is a file where you can see this is the result, where you can see this is the actually result. So likewise, you can see in your database sequences total 11 sequences, and uh, your query sequence is this, which aligned with sequences. In this case, there is no hit found for query sequence, and for other sequence. So actually, there are not much similarity exist in the sequences. So that's why there is no hit found like case. So in our case, in another case, you can see when sequence similarity is significant among sequences, then the result will appear like this, like in a database which we have used prior, there are 9 sequences, and in query, what is the query name? That is this one, and total significant aligned with this database sequence. And now you can see, as we have explored this previous, so this is the result. Uh, so here I have another example, how to save the result in Excel format, which is easy to sort the result. So you can check, you just uh, put your query name. I am using another example because in this case there is no significant hit found. So I am using another case where I have stored the query sequence in the test.fasta, name of the test.fasta, and the database sequence is the database tvsic.fasta, and out uh, result 2 dot out, and here I am specifying the output format, that is out fmt is equal to 6. So this is a specific file format. Now you will obtain the result in a Excel file format, looking like Excel format. You can easily sort the data accordingly. So just click okay. So just click on this and you can see there is a file name, that is final result 2.out. Final result 2.out. Okay. You just open this file in Excel. But Excel is not in there, so we can use to open it in Excel like this way. Okay. So here you can see your query sequence 1 aligned with your database sequence, this one, with 98.8 percentage identity and 0 e-value at 1770 bit score. So these three, which are very important parameter, on the basis of this you can select, you can say that query 1 is most similar sequence with this one. Likewise, in query 2, it also shows the sequence similarity 98.345% with 0 e-value and 1687. And in case of queries 2, also aligned with other part of the sequence at this location at 86%. So you can see the alignment of the query start from 1 to 988, and in other case start from the alignment of the query from 1 to 967. So I think this cover most of the query sequence, that is known as query coverage. So these are the parameters. On this on the basis of this you can sort your results. Okay, so likewise you store this database, this result, and you can put your header on that file from here you can paste like first one is the query sequence id, subject sequence id, percentage, identity, length, mismatch, gap, whatever. You can just paste these headers on your uh, excel file here by creating just inserting one row, just one row, and you just paste all the headers. That will easy to understand to other ones to how, what is the result actually. So after that you can save whatever place you want. You can save this. Okay, so these are the basic steps to run basic local alignment search tool on your own computer.